Okay, then we have here the chemical method for your sterilization or your disinfection. So we classify here your chemicals as either antiseptics. So pag antiseptic, sa skin lang natin siya ina-apply. This includes here your number 1, alcohol. Alcohol here includes your ethanol and your isopropyl alcohol. Basically, try to kill your microorganism by disrupting here the protein, try to denature the protein structure of the membrane of your bacteria. Next one, we have your halogen in the form of your 2% iodophore, which also have inactivate the proteins. Then we have also here your heavy metals in the form of your silver nitrate and your mercury chloride, which also try to disrupt here our protein structure. Then we have also here the phenol in the form of your chlorhexidine gluconate chloroxylenol, which also try to disrupt here the protein structure of your bacteria. The following here, on the other hand, are used as disinfectant. So, pag disinfectant, so nasa surface natin siya, hindi natin siya ina-apply ulit sa skin natin, di ba? So, the first one, we have your aldehyde group or aldehyde group of your chemicals includes your formaldehyde, formalin, at a concentration of 1 to 8%, and the 2% glutaraldehyde. So, this one try to kill your microorganisms primarily by disrupting the amino group, the carboxyl group, and we have the sulfhydryl group of the protein structure of the bacteria or your microorganisms. Halogens includes your chlorine compound like your bleach. Okay, it's also react to the water to form here your hypochlorous acid. So hypochlorous acid is toxic to your bacterial cell. Detergent in the form of your quaternary ammonium compound will also try to disrupt here the protein, the cell membrane of your microorganisms. And the phenol here na disinfectant, we have your hexachlorophene. Okay, so it's able to kill your microorganism by denaturing the protein structure of the membrane of your microorganism. Okay, then we have also here your chemicals and gases here which are considered to be sterilizers. So your aldehyde could also be act here as sterilizer. So we have that in the form formaldehyde, the 6 to 8%. Kanina, di ba, ang disinfectant natin na formaldehyde is only 1 to 8%. This one, 6 to 8%. Glutaraldehyde is also 2%. We try to disrupt here try to disrupt the membrane, amino group, sulfhydryl group, and carboxyl group of your bacteria. Okay, so the manner of the contact time, like if you use here your glutaraldehyde as your disinfectant, so medyo mas matagal dapat ang contact time natin, exposure time ng materials mo na sinisterilize if that one is sterilizer ang gamit ng glutaraldehyde compared if that one is just a disinfectant. Gases, we have your ethylene oxide, halogen peroxide, periacetic acid. Responsible here for alkylation of the nucleic acid and therefore disrupting the or killing the entire bacterial cell or other microorganisms. Okay, then we have here some of the guidelines for safety program, the clinical lab. So first one, so mga basic lang to, hand washing. So wear gloves, PPE program. So sharps would eventually be disposed in the puncture-proof container and some environmental control. We have also work practice, so we are observing it naman, so do not eat smoke inside the laboratory. Then disinfect your working area before and after doing the laboratory activity. No recapping of your contaminated needles. So try to dispose of one in your puncture-proof container. PPE program, so we have here to wear your gloves, your mask, your lab gown, face shield. And we have your safety from infectious agents. So mucous membrane contact is the way of transmitting the infection here. So when you are rubbing your hand or even your nose, that's why you need to wash your hands always. Okay, another route here of the transmission of infection is the airborne. So airborne, most likely here because of respiratory droplets. So like your Mycobacterium tuberculosis, your Brucella, and also your Francisella. Another one, ingestion, so food and mouth. Fecal oral, just in case of your Salmonella and your Shigella. And we have direct inoculation through your accidental needle stick puncture, even your blood transfusion, mga blood-borne ito, like hepatitis A, hepatitis B, hepatitis C, D, and your HIV. We have here the most frequently identified infection in the laboratory, or most frequently identified infection in different workplace. The first one, Salmonella and Shigella. So basically that one is by ingestion process. Mycobacterium tuberculosis, brucellosis through your inhalation. And we have also your hepatitis through your direct inoculation. Okay, now we discuss about the different biological safety cabinet or hoods. So basically your biological safety cabinet or hood here is a containment barrier that would protect here the personnel, laboratory personnel, when we are processing our specimen in the microbiome. So the class 1 is characterized here, the room air, okay, is passed through your cabinet, okay, then you try to circulate on that, lalabas, okay, pupunta sa HEPA filter natin, eventually is being exhausted. So ito, open ang kanyang front, so papasok ang air dito from the environment, okay, circulate siya sa working area, dadaan sa HEPA filter, filter siya palabas. Again, air from the environment, papasok sa workplace or work area, dadaan sa HEPA filter, then lalabas. Okay, the class 2 biological safety cabinet natin hence the most commonly used na biological safety cabinet laboratory. So this characterized by open ang kayang front then. Only na pinagkaiba niya, air is pulled inward and upward through your blower, passes through your HEPA filter, bago siya pumunta sa work area, ang air, and eventually pass out, dadaan ulit sa HEPA filter, and eventually exhausted to the environment. So, anong biological safety cabinet 2 natin? So, the air here from the environment pa din siya. Papasok dito. Bago siya pumasok, after niya pumasok, dadaan mo na siya sa HEPA filter bago pumunta sa workplace or the work area kung saan ka nagpa-process. Okay, then babalik sa HEPA filter and lalabas. Ano pinagkaiba nito sa class 1? Class 1, papasok ang air, okay, dadaan sa work area mo bago siya i-filter. Dadaan sa work area, pupunta sa HEPA filter, and lalabas. Your class 2 naman, papasok, dadaan muna sa HEPA filter, pupunta sa work area, dadaan sa HEPA filter, palabas ulit. Okay, the next one, we have your biological safety cabinet 3. So this is a self-contained ventilated system with the front here, meron siyang gloves. So self-contained mean to say the air, hindi siya galing sa labas. Dito siya mismo, it's being supplied and circulated within the entire system or nasa loob siya. So walang air na pumapasok dito coming from the environment. All you need to do is just insert your hand in the gloves for processing, manipulation of your sample or specimen inside this one. So this is used primarily for highly infectious na mga biological agents. Okay, now we have here the biosafety level. So we have here the biosafety level 1 is when we are encountering pathogens which are not consistently causing infection among healthy adult populations. So this one, pwede ka mag-work lang para sa table lang, within the table, ganon. Sa bench top, ganon. So this one includes your Mycobacterium gordonii, Bacillus subtilis. It might require here biological safety cabinet level 1 lang. Or pwede yun sa bench ka mag-work. Another one, we have here your biosafety level 2 is characterized by those pathogens which are commonly encountered or usually causing infection. Okay, so this includes your Staphylococcus, hepatitis B virus, HIV, enterics, mga gram-negative bacilli, and we have your Yersinia pestis and your Bacillus anthracis. So you work here under your biological safety level 1 and your biological safety cabinet level 2. Okay, level 3, on the other hand, is for those when you are handling microorganisms which are uncommon. Hindi siya common. And at the same time, this one is transmitted primarily by aerosol. So, medyo toxic ito. So, require here biological safety cabinet 1 and 2, but most likely dapat nasa 2. Okay, so you have your ducted air ventilation special PPE program, okay, for the personnel. But this includes here, you like your Francisella, Brucella, Mycobacterium tuberculosis, systemic fungi, and basically they're transmitted by aerosol. Okay, level 4 na biosafety, itong mga toxic na mga nakakatakot na mga biological agents. So these are potentially toxic and exotic na microorganisms transmitted primarily by aerosol or airborne or even mga novel na mga pathogen. So hindi natin alam or unknown ang kanilang route of the transmission. So example for that, your Ebola virus, okay, your Lassa virus, SARS, your SARS-CoV natin, SARS-CoV-2 or nCoV natin, dapat dito din siya sa biosafety level 4 dapat siya. Okay, so require here biological safety cabinet level 3 kasi dapat closed system ito. And then the laboratory handling this type of the specimen or this type microorganism should be situated in a separate laboratory or separate building. Tapos meron ka dapat na changing room. Okay, so highly controlled, restricted ang area na to. So dapat before you try to enter this type of the laboratory, okay, or type of the lab, we try to handle this biosafety level 4, dapat may changing room, okay, and then we'll try to change bago ka pumasok, and mag-change ka ulit bago ka lumabas. Because this is highly contagious and infectious na microorganisms. Okay, thank you.